ESF is the oldest, largest, and most distinguished institution in the United States that is focused on the study of natural resources and the environment. Hello and welcome to this edition of Improve Your World with SUNY-ESF. I'm Dave White. Today's program focuses on research efforts to bring back the American chestnut tree, a tree devastated by blight at the turn of the last century. Scientists believe they are very close to a solution.
We've been working on this for a long time. We've been studying this one particular gene, the oxalate-oxidase gene, and over the years it's really convinced me that this is the gene that's going to really do the trick. For one thing, we've put this gene into hybrid poplar over 10 years ago, and we've shown in transgenic hybrid poplar that we can enhance pathogen resistance in that tree. That was to a different pathogen, but it's still giving us evidence that it would work in our tree also.
Well, people have been working, ever since the discovery of the blight, we're about the third generation of scientists that have spent their careers investing in figuring out some way to get around the blight, avoid the blight, find something that was resistant to the blight, or some way solve the problem. And so we're hoping that ours is the generation that will succeed. This
was a key species in eastern forests. It was super productive at producing nuts for wildlife, very important for agriculture, for human consumption of the nuts, very important for the lumber industry, making a very rot-resistant, fast-growing wood product.
First off, 1 out of 4 trees in the eastern United States was the American chestnut in 1904 when the blight was discovered. So it was a hugely, from physical presence, it was a huge tree, dominated the overstory in most of the eastern United States, especially in the Appalachian range.
In fact, in the Appalachia, people used to release their hogs in the forest in the fall to fatten up for the wintertime.
It was important for the nut crop, was important for wildlife. Many wildlife populations crashed after the chestnut disappeared out of the forest. It was a very reliable species. It produces a nut crop almost every season. Oak trees and other things that came in to replace it were more periodic. They would have a nut crop every 3 or 4 years. And that's not good if you're a wildlife going into the winter. So it was tremendously important for wildlife. It was tremendously important for the people in the region.
American chestnut has been immortalized in song, chestnuts roasting on an open fire, or the village blacksmith, under the spreading chestnut tree, the village blacksmith stands. In books and stuff, Walden, Thoreau's book, he mentions chestnuts in there, and it's a very nice passage in there about American chestnut. So we really want to bring it back, and the only way it can come back is to make a resistant tree, because you cannot control blight any other way but making a resistant tree. And so once we have a resistant tree, we'll start a major restoration program, and it'll take time. It's going to actually take probably over 100 years to get it really established throughout its natural range. But we have to start somewhere, and that's where we are right now, at the beginnings.
How did the American chestnut tree get into this position? It's a tale often told about a foreign species of plant brought into the country by accident or before people understood the dreadful unintended consequences.
We don't have the chestnut the way we used to because of an exotic pathogen that was introduced into this country about a little over 100 years ago. And when that came in, it was brought in on Asian species of the chestnut. And our American chestnut was not resistant at all to this particular disease. And so it quickly swept through the forests, spreading within 50 years, killing over 3 billion mature American chestnut trees. And today, American chestnut's pretty much just surviving as a small understory tree, a few million left out of the billions that there once was. And we would like to restore it back to its grandeur. So this is a culture of Cryphonecter parasitica. It's the fungus that causes chestnut blight, and it's the fungus that was brought in from Asia and is basically wiping out the American chestnut trees. This fungus will enter a wound in the bark of the chestnut. It will colonize that wound and eventually start producing oxalic acid and mycelial fans. And once those are formed, it starts invading the healthy tissue of the chestnut, forming what's called a canker. This is an example of the problem with the chestnut blight. These are cankers formed by the fungus. They enter through a wound and eventually form this canker. These things eventually will grow and girdle the tree, killing everything above it. Meanwhile, the conidia, or the spores, spread down the tree, form other cankers, till finally the base gets girdled and kills the whole tree above the ground. And basically that's how chestnuts are surviving today at the ground level. They will send up new sprouts, but if you can see even here, they get little cankers on them, and those eventually die too. That's why you can't find big chestnuts anymore. None of the big 8 to 10 foot diameter trees anymore. It's all little trees like this.
Reiterating something Dr. Powell said earlier, the only way to bring back the American chestnut tree is to make one that's blight resistant, because you cannot control blight any other way. So the only way back into the forest is through several different laboratories.
First of all, we have to find some kind of gene that will confer resistance to the chestnut blight. Because the American chestnut tree has no resistance to blight. It never co-evolved with this, has no way to defend itself. So we've looked at many different sources of genes to put into the chestnut. One is from other plants. For example, we get one of our first genes from wheat. It encodes an enzyme called oxalate oxidase. And basically this gene will detoxify or break down an acid that the fungus throws at the plant. Okay, so the way this fungus attacks a plant, it produces oxalic acid, and that oxalic acid basically kills the plant cells. So we want to give the plant a defense against this acid. And we're not the only ones who are doing this. We're trying to put this into American chestnut. But a lot of other researchers are interested in putting this into other crops, such as peanut. And they're using that to fight off diseases of peanut. People also put it into soybean. People are putting it into sunflower. People are putting it into tomato. And of course, we are putting it into chestnut. More recently, we started getting genes from Chinese chestnut. Now, Chinese chestnut is from a part of the world where this disease is endemic, or is natural. And so the Chinese chestnut has co-evolved with this pathogen, and therefore developed resistance on its own over long periods of time. So we are now just recently going into the Chinese chestnut genome and trying to pull out the genes that allow it to be resistant to chestnut blight. This is not an easy project to do, but we are looking at several genes right now. We have about 30 of them from the Chinese chestnut that we are putting into American, and we're going to see if we can confer resistance with those genes. A lot of times people ask, how do you get a gene into a plant? We don't directly put the gene into the plant. We actually have a natural genetic engineer that helps us. It's called Agrobacterium. A culture of Agrobacterium tumefaciens. It's a natural genetic engineer, and it's because of this we can put genes into the American chestnut. This particular bacteria naturally puts genes into plants. Usually in the wild it does the genes it wants to put in, but what we have done, and other scientists have done, is to put the genes we wanted to put into the chestnut tree into it, and then expose the chestnut cells to this bacteria, and it will put it in for us. Right now she is transferring some embryos from American chestnut. These embryos are what we actually transform or put the gene into when we're putting genes into chestnut. And once we get them into these embryos, we have to go through a long process, which you'll be seeing soon, to get these embryos back into whole plants. But this is basically the starting point.
Using this microscope, we can have a good look at chestnut embryos that have had the enzyme from wheat genetically implanted.
So what we have here is the incubator containing our embryos. These different racks represent different transformation experiments. And as you can see, we have a lot of them here. All these things are in the very early stages of the pipeline for developing transgenic chestnut trees. All these will have to be regenerated into whole plants. So we see a lot of them here. We have right now what's called 130 events in here. Each of those are going to be a different line of trees.
In this lab, we're responsible for getting from a little tiny embryo in a petri dish, a little green embryo, into a whole plant ready to go out into the field. And so it's the embryos, when they come out of Bill's lab, you got a little, they're tiny little things. They're about the size of a pencil point, and many of them just refuse to regenerate into a whole plant. So we have maybe out of 200 that come over from Bill's lab, we get maybe one shoot to form. So it takes a lot of embryos. So each of the different cell lines that he produces, he gives us maybe 3 or 4 plates of embryos. We multiply them up from there, and then we put them into a different set of growth regulators and a different media, and we regenerate from that whole plant. Let's see, first we get shoots, and we get multiply up the shoots until we got hundreds and hundreds of shoots, and then we root those, and then we put those into the growth chamber and eventually out into the greenhouse. It takes just about between the time it comes out of Bill's lab and we're ready to go into the field, it's just about a little over a year. This is what comes over from Dr. Powell's molecular biology lab. We get a little plate full of embryos that have got the new genes put in them, and the next step is we need to we put them in, transfer them into a different media that encourages shoot production. And if you notice down here in this one little corner, there is one little shoot coming up out of all of these embryos. Hundreds and hundreds of these embryos out of a petri dish like this, we might get one shoot, but that's all we need, because once we've got one shoot, we're off and running.
So this is where we do our propagation. This is what we use to grow our plants in. It's a gel, kind of like Jell-O, with sugar and minerals and nutrients, everything that a plant needs to grow, without soil. What we do every month or every 3 weeks is take these cubes of plants. I'm going to pull a few out. This is a little baby chestnut. Okay. Cut off some of the big leaves. Cut off this base. This is callus. This is undifferentiated plant tissue. We don't want that. We just want the shoots. So we've got a little one over here. Cut off a couple more of these big leaves. Here's one right here. And there's another one. So in this case, from one shoot, kind of on the smaller side, I got 3 new pieces which will all grow up to be nice big shoots, from which I can chop all of those into more pieces. When we get these nice big shoots, the other thing we can do is start them getting, convince them to get some roots. So it starts out the same, where we cut off the base of callus at the bottom. Then we take the leaves off of the bottom third of the plant and make a little diagonal slice and place it in this rooting hormone, which is called IBA for short. This is a plant hormone that stimulates root growth. Let them sit in the hormone for about a minute. And then we use this special media here, which has a little bit of activated charcoal in it, which shades the roots and stimulates root growth as well. Then we take these and gently put them in the rooting media.
Now this is about a week to 10 days later. They've come out of the rooting and they've gone into this post-rooting media, and they've been doing very nicely. This is about a week later. You can see all the beautiful roots on the bottom. It took us years to be able to do that, and Allison is a master at it.
So I have a little magenta cube with our TC plants, and I'm going to put them into potting soil. And I usually want to try to find one that has an alive shoot tip. Let's take this one. So I just gently pull it out like that. And then I'm going to make a little hole in the potting soil. And gently pack it down around the roots. So it looks like that. Just going to water it a little bit.
You might think the hard part is over. The tiny chestnut embryos are now tiny plants. They have roots. They are ready to grow into trees. Not quite. The plants need a big assist, and that's delivered in the Equalization Laboratory.
We've had this room for about since 2007, It was purchased, we split it with the 50-50 with the American Chestnut Foundation, and the college picked up the tab for the other half. And it's just been a lifesaver. Before we had the facilities, I'll show you inside, we were killing off better than 95% of our plants before they got out to the field. We had roots on the bottom, we had little shoots, we'd work so terribly hard, but we'd kill off 95% of them on the way out the door. And so the Chestnut Foundation, the president of the Chestnut Foundation came to me and said, Chuck, what can we do to help? And I said, well, you can buy us a couple of growth chambers. And he said, how much? And I said, oh, about $50,000 a pop. And he sort of blinked, but within 6 months he had the money, and I had we got the access to the room, and we put it together, and we've had this ever since, and it made a tremendous difference.
So, problem solved. Not quite. Let's go back to lab technician Logan Will and watch her finish preparing the plants for the growth room. She puts a plastic bag over the baby plant and anchors it with a rubber band, which, it turns out, helps the program take a significant leap forward.
So that's it.
The biggest problem with getting a tissue culture plant out to ready to go outside is that the foliage, the leaves, are completely worthless. The stomates are stuck in the open position. The cuticle layer is absent. So a tissue culture plant will go from perfectly healthy inside the cube where you've seen it to dead as a stone within 20 minutes. So we have to have some way of keeping it very, very moist. So what I did, I called up all the growth chamber manufacturers and said, when I open these doors, I want to see fog roll out. They all told me they couldn't do that, so we finally agreed. One growth chamber manufacturer said he would strap on the biggest humidifier unit they make into a medium-sized chamber. He'd never done it before, but he sold us that way. And he thought he could get us 95% humidity. Now that's very, very humid. If you go out on a summer day and it's 95%, you are going to die. It's so humid. But if you're a little tissue culture plant, it still wasn't enough. So we had spent $50,000 each on these chambers, and we still were killing off maybe 50, 75, 80% of the plants. So then somebody, we don't know who did, nobody can remember who came up with the bright idea, but so we spent $50,000 to get up to 95%. And then we spent another nickel and another nickel on a rubber band and a Ziploc baggie to get up to high enough to where we could keep the plants alive. So that's what we do in here. We put each of the little plants before it goes into the pot, gets one of these little gets a little plastic bag and a rubber band. And that made all the difference. And with this treatment we can get probably 50 to 75% survival into the greenhouse, which is where we'll go next.
All right, we're in the ESF greenhouse. This is the next stop after the growth chamber. So trees come out of the growth chamber, tiny, inch or so tall, tiny leaves, and this is kind of the next step in acclimatization where they're starting to look more like real trees, what we recognize as trees. So after they've been in here a couple months, they'll start getting bigger leaves, start getting a little taller, and in another couple months we'll be able to plant them outside.
So if you look over here, these trees that are planted close together in the tree tubes, this is our efforts to figure out how to upscale production of these trees. This is a simulated seed bed, even though these do come from tissue culture, not from seed. Planting them on very close intervals, and during the summer we actually have a shade tent that goes over top that. So every tube has a tree in it. Tubes help them to start off nice and straight and protects them from rodents and things like that. So these are the trees that come right out of the greenhouse, grow for a season or two in closed quarters, and then we would dig them up, probably bare root them and move them to other places. In fact, I think a couple of these trees in this will be the ones that go to the New York Botanical Garden.
That's right. Dr. Powell said transgenic American chestnut trees are planted in New York City. We'll get to that in a moment. But first, a little more on the science at work here.
The first step of the RNA extraction is to grind up the plant tissue to a fine powder, and we do that using a mortar and pestle and liquid nitrogen. And you can see we have the leaf is all ground up to a nice fine powder.
In this procedure, lab technician Kathleen Baer is extracting RNA from genetically modified shoots so their gene expression can be examined. The powder is put in a solution and the RNA extracted for analysis.
And this is one of the ways we analyze that RNA, to look at the genes that we've put in and how they're actually responding inside the plant. Put the RNA in this machine, and this display shows different amounts of expression.
In other words, how effective is each gene at affecting changes in the new chestnut tree? Farther along in the growing process, a leaf assay is performed.
This is a way to predict how the trees are going to react to the chestnut blight, but a test that we can do earlier on in the process. So instead of inoculating a tree when it's maybe 5 years old outside, we can take a leaf off a tree that's much younger and test it with some of the blight fungus to see how it's going to respond. I'm going to start by making a wound on the mid-vein of the leaf. I'm going to use a scalpel and just make a tiny slit along the mid-vein here. This simulates a wound that would happen on the bark of the tree. Next, I'm going to take a plug of the blight fungus that we've cultured in the lab. So I have a little round plug here that I just cut, and I'm going to put that so the fungus is right against the wound. And then after the leaf has been exposed to the fungus like this, we'll put it in a tray that'll keep it moist. Cover that, seal it up, and store it in the dark for about 1 week. After this week, there will be staining that will appear next to the wound that I created, and based on how much of the leaf is stained, we'll be able to predict how that tree will respond to the blight in more natural situations.
Now, just recently, we had some transgenic trees made that produce large amounts of this enzyme. And what we found is if you reach a certain point with this enzyme, you get a great inhibition of the fungus on our leaf assays. Okay, so we have at least 5 lines right now or more that are looking very, very good for resistance. Now, saying that, the caveat is that we don't know for sure until these trees reach 3 to 4 years old. That's when we will test them, and they might be fully resistant or they might be partially resistant. If they're partially resistant, the great thing is that we are looking at lots of other genes that we can stack with this oxide oxidase gene, and with the combinations we might reach the full resistance and have a restoration tree. But it could be this gene by itself. Well, we will see.
They were confident enough in these 5 lines of trees to accept an invitation from the New York Botanical Garden in New York City to put in a test plot in a very significant location.
We're very excited to be going back to the New York Botanical Garden because that's a stone's throw, right literally across the street from where the blight was discovered in 1904. And so it's just such a full circle effect that it's just very exciting to us.
It's a chance for Doctors Powell and Maynard to update many of the people and organizations that have helped fund the research.
Many of you were involved with this project from the get-go. Many of you made major contributions along the way. This is like old homework. This is just great.
Art of genetic engineering was very new when we started. There was only a handful of plants that had been genetically engineered, and only 1 tree.
Following their lecture, everyone gathered at the test site to plant the last of the 10 trees at this site, and a chance for some of the laboratory staff to join in the celebration.
The building evidence over time has finally got to a point where I think we can be pretty confident with these trees. Again, I can't guarantee it, but I think we have high confidence that these will, well, I'm almost certain that these will have enhanced resistance. The question is, will it be a high enough resistance that we want to use in a restoration program? If it's high enough, we will immediately go into that. If it's not high enough, we will continue to add additional genes until we get to the level that it can go into restoration.
That's it for this edition of Improve Your World with SUNY-ESF. Please join us again next time.